Formation of ATP. Ever question yourself how you can move your body, like exercising, running, or even dancing? And how do I have such capabilities to do so? What's the main purpose of eating? Why do I have to eat to stay active? Well, it's all start with a small, relatively simple molecule called adenosine triphosphate, or also known as ATP. These molecules are often referred as the currency of biological energy or body fuel. Adenosine triphosphate is made up of a sugar called ribose, with nitrogenous base called adenine, and 3-phosphate group attached to it. The energy released by kicking one of the phosphate group off is used to power many energy-requiring cellular reaction. Well, this is how the ATP is spent. Our body derive energy from the food we eat, particularly from glucose, through cellular respiration. In order to be able to use it, our cells needs energy to be transferred into specific form of stored energy, ATP. To turn glucose into energy, oxygen is required. Glucose and oxygen will be converted into 6 molecules of CO2, 6 molecules of water, and some energy through cellular respiration. But the question here is, how exactly glucose is used to produce ATP? In short, the process has 3 stages, which are glycolysis, followed by Krebs cycle, and lastly oxidative phosphorylation. The first stage is glycolysis. Glycolysis is the process that extracts energy from one glucose molecule, which is broken down to form 2 molecules of pyruvic acid, also called pyruvate. Glycolysis is an ancient metabolic pathway that has evolved since long ago and can be found in most of living organisms today. Glycolysis occurs in the cytoplasm of animal cells, plant cells, and the cells of microorganisms. This process can be broken down into 2 main phases. In energy-requiring phase, the molecule of glucose are rearranged with 2 phosphate groups attached to it. The phosphate groups will drive the modified sugar, fructose 1,6-biphosphate, to become unstable and divide in half and form 2 phosphate-bearing 3-carbon sugars. The 3 carbon sugars that were formed earlier, DHAP and glyceraldehyde 3-phosphate, are different from each other as they are isomers. Only glyceraldehyde 3-phosphate can proceed with the following steps of glycolysis. Although DHAP will eventually be converted into glyceraldehyde 3-phosphate. In the second phase, each 3-carbon sugar is converted into another 3-carbon molecule, pyruvate, by a cascade of reactions in which 2 ATP molecules and 1 NADH molecule are created. This phase will take place twice, once for each of the 2 3-carbon sugars. Overall, the process of glycolysis converts 1 6-carbon molecule of glucose into 2 3-carbon molecules of pyruvate. 4 ATP molecules are synthesized and 2 ATP molecules are used during glycolysis, for a net gain of 2 ATP molecules and 2 NADH molecules. The second stage, Krebs cycle. What is the definition of Krebs cycle? Krebs cycle is defined as the second stage of cellular respiration, and it is also called the citric acid cycle. It is known as the Krebs cycle after Sir Hans Adolf Krebs, who discovered its steps in 1937. ATP is needed to produce energy. Pyruvate oxidation. Pyruvate oxidation process happened right before Krebs cycle process in mitochondrial matrix. In this process, pyruvate is converted into acetyl-CoA. Acetyl-CoA is important substance required to start Krebs cycle. Krebs cycle. Krebs cycle process is a series of chemical reactions that is used by aerobic organisms to release stored energy through the oxidation of acetyl-CoA, derived from carbohydrates, proteins, or fats, into ATP and carbon dioxide. There are a total of 8 basic chemical reactions that is carried in this process. Krebs cycle started when acetyl-CoA combine with oxaloacetate and water. This cycle will be continued until there is no supply of glucose to produce pyruvate. Hence, no energy will be produced. The end product of Krebs cycle for one glucose cycle are 8 NADH, 2 FADH, and 2 ATP.
The final stage, oxidative phosphorylation. Oxidative phosphorylation is made up of two closely connected components, which are the electron transport chain and chemiosmosis. But what is electron transport chain and chemiosmosis? Electron transport chain is a series of proteins and organic molecules found in the inner membrane of mitochondria. Electrons are passed in a series of redox reactions. Energy released in these reactions is captured as proton gradient, which is then used to make ATP in a process called chemiosmosis. Diagram shows the major components involved in oxidative phosphorylation. The process starts with NADH produced during earlier stages. NADH is oxidized to an Ad+, thus release electron and H+. Electron is transferred to complex 1. Meanwhile H+ is pumped into intermembrane space, and there will be an accumulation of H+ in the intermembrane space. Thus, pitot gradient begins to form in the intermembrane space because it have greater H+ than in the mitochondrial matrix. Electron from complex 1 is passed into coenzyme Q. FADH2 give up its electrons and turn into FID. It donates its electron to complex 2. Then, electron from complex 2 is passed into coenzyme Q. Coenzyme Q is reduced and passed the electrons to complex 3. As electrons move through complex 3, more H+ are pumped across the membrane. Complex 3 reduced and passed the electrons to cytochrome C. Then, cytochrome C reduced and passed the electrons to complex 4, where a final batch of H+ are pumped across the membrane. At this point, massive proton gradient has formed. Complex 4 passes the electrons to oxygen, which is the final electron acceptor. Then, it splits into 2 oxygen atoms and accepts proton from the matrix to form 2 water molecules. ATP synthase uses the energy stored in the form of proton gradient and phosphorylate ADP into ATP. Total ATP, NADH and FADH2 produced from the glycolysis and Krebs cycle processes are shown. However, each NADH produces 3 ATP, meanwhile each FADH2 produces 2 ATP. Therefore, NADH produced 30 ATP, meanwhile FADH2 produced 4 ATP. The total ATP produced via oxidative phosphorylation is 34 ATP, and the total ATP produced in substrate-level phosphorylation is 4 ATP. Total production of ATP per 1 glucose molecule is 38 ATP.